last speaker is Alyssa, also from the Parsec Lab, talking again about Pseudomonas, but something different.
Thank you for sticking around. I'm Alyssa. Today I'll be presenting the work I've been doing to identify protein interactions in the CPX system of Pseudomonas aeruginosa. As mentioned previously, Pseudomonas is an opportunistic pathogen. That means it's found in the environment, like in soil or in water, but it also has a clinical significance in the infection of immunocompromised individuals. It is a gram-negative bacteria, which means it has an inner and an outer membrane. These two membranes, as well as a layer of peptidoglycan, constitute the cell envelope. The cell envelope is the first layer of protection for these bacteria, and it's surveilled by a network of various stress response systems, which allow the cell to identify and respond to various environmental stresses. In these cell envelope stress systems, mutations can arise spontaneously during host infection. For instance, here is CPXS. It's an envelope stress sensor protein in the CPX system of Pseudomonas aeruginosa. And in this protein, in populations of Pseudomonas that have either evolved under antibiotic stress or that have been isolated from chronic host infections, there have been many mutations that have been identified. And these mutations are significant in the CPX system of Pseudomonas in its ability to infect the host. And that leads to a question of what is the CPX system in Pseudomonas aeruginosa? What does it do? And why is it important for pathogen adaptation? Moving in the direction of trying to answer this question, it's important to first characterize the system. So here it is. This is the CPX system. It's a two-component system. It's been very well studied in E. coli. And from research with that bacteria, it's known that there are two core proteins, CPXS and CPXR. S is a sensor kinase that transfers a phosphate group to R, and that phosphate transfer is what activates the system. There's also a negative regulator key. And as I mentioned, Pseudomonas aeruginosa does have its own CPX system, though there are also two novel accessory proteins, 3203 and 3207. 3203 and CPXS are predicted to be membrane-bound, whereas R and 07 are cytoplasmic. The CPX system in Pseudomonas is largely uncharacterized, and thus the functions of 03 and 07 are unknown. We do, however, have AlphaFold models that predict that 3203, 3207 interact with CPXS and R. So I am framing my research around validating these predictions. To do so, I am using the bacterial-2 hybrid assay. This utilizes two adenylate cyclase domains, T18 and T25. When these domains are brought into proximity, we get cyclic AMP production, and when they're separated, we don't see any production. This is a calorimetric assay, which means that our interactions are represented by a visual readout. So for a bacteria that has cyclic AMP production, when we plate those on indicator plates that have a pH indicator and maltose, we see this nice, pretty pink color. This is a result of maltose fermentation, which is induced by cyclic AMP. And when there isn't cyclic AMP, we don't see any maltose fermentation from the bacteria, and thus there's no colony color change. For the methodology of my experiment, I am taking a protein of interest, fusing an adenylate cyclase domain to it, and I'm transforming two plasmids with these protein fusions into an E. coli strain that lacks an adenylate cyclase. If the proteins interact and the domains are brought into proximity, we get that color change. If the proteins either don't interact or the domains are in an orientation that doesn't allow them to come into contact, we don't get cyclic AMP production and there's no change in color. With that being said, we do see a gradient of positive interactions with this assay. So all of these are indicative of a protein interaction. It's just something to keep in mind when we're looking at these experimental samples. Here's my first one. This is CPXS and R. This is an expected interaction. They're the two core components of the system. can see we have two different results, one positive and one negative. As I mentioned previously, this is likely due to that orientation difference between these fusions. To elaborate, let's look at the positive first. So we have a C-terminal fusion of CPXS and an N-terminal fusion of CPXR. This orientation allows those adenylate cyclase domains to be brought into proximity, so we get that color change. However, when we have fusions in this orientation where the C-terminus of R is fused and the N-terminus of CPXS is fused, those domains aren't being brought into proximity, so we don't see anything. With that being said, because we get one positive interaction, that is indicative of a protein interaction. Moving on to my other experimental samples, this is 3203 and CPXS. In this orientation here, where our 3203 tag is up in the periplasm and our CPXS tag is in the cytosol, we don't see any interaction. However, when that orientation changes with the fusions, we do get a response. This is 3203 and CPXR. We have a periplasmic tag up here on 3203 and a cytosolic tag on CPXR. If you recall from that interaction between S and 3203, where there was a periplasmic and a cytosolic tag, we didn't have an interaction. That's not what we see here. And this is also not expected. This assay is tricky, and there can be artifacts of protein overexpression. So it is possible that there's an overexpression of 3203, which is causing it to be expressed in the cytoplasm. However, I plan to continue looking at this interaction, and in this orientation, we don't see an interaction when they're both in the cytosol. Here is 3207 and CPXS. We don't see an interaction here in this orientation. However, when that tag on 3207 is switched to the N-terminus, we do see those domains coming into proximity. And lastly, here is an interaction between 3203 and CPXR. We have a lovely bright pink colony here with an N-terminal fusion of 3207 and an N-terminal fusion of CPXR. One more thing I would like to point out is that here on CPXR, that adenylate cyclase domain is tagged near the phosphate transfer portion of the protein. This is a highly active area, and so that could be influencing where or which orientation those domains are coming into proximity. Because when we move that tag away from the phosphate transfer domain, we don't see an interaction. The work that I've done with the bacterial 2 hybrid assay suggests interactions with 3203 and 3207, CPXS and CPXR. And in the future, I would like to continue exploring these interactions between the CPX proteins. I'm hoping to collect more quantitative data so I can measure the strengths of these interactions. And I'm also hoping to explore the biological significance of these two novel proteins, O3 and O7, by effectively using them as bait, so I can see where else they might be interacting outside of the CPX system. I'll be using techniques such as pull-down assays and proximity labeling for that. And of course, I would like to thank my lab, Matthew Parsek, for giving me this opportunity, Haley Deering, who's been so wonderful in mentoring me, and also Megan O'Malley for also mentoring me, and just everybody's so wonderful. Thank you for also being here, everyone. Wow. And I look forward to answering any questions that you have.
Do we have a burning question? Okay. With the 3203 protein, is it possible that it is inverted from what you're showing? Or is that just completely impossible?
That is definitely possible. This model here is based on predictions that were from this assay is kind of tricky with membrane proteins too, so it's kind of hard to say, but it's definitely possible. Yes.
So I guess on that note, you said that, I believe you said you think those dots are like within, reactions happening within the cytoplasm, right? If I recall correctly.
We don't know, it could, I'm thinking it's due to overexpression, so it's not actually reaching the membrane, and it's instead in the cytoplasm.
Okay, I was just curious if maybe it could be that the dots you're seeing are just like the top of the membrane, or
The top of the membrane is in the cellular membrane? Or
yeah, just like this, the 3D perimeter versus inside.
It is possible. The best answer I have is that these bacteria are extremely small, and so this schematic here is mostly for us to be able to visualize what could be going on, but it likely looks very, very different in the bacteria. And things move around a lot too. But I'm hoping that when using like other techniques that we can purify the proteins to see what they interact with instead of having, I'm not sure if that answers your question.
I think it does.
Yes.
I can't remember, did you say at the beginning that 3203 or 3207 had phenotypes? Like when you were talking about, if you make mutations, do they have phenotypes?
I did not mention that. I don't know. I haven't done any work with knockdowns with these proteins or in general.
Like, just to see if they have an effect on CPXR activity.
Yes.
You know, like a gene that would be turned on or off.
We have been, that is actually in the works. We're looking at like constitutive on CPXR or S to see if we still get interactions with these proteins. So, to be determined.
Okay.
That was my question too.
Okay.
I was gonna be more eloquent, but.
3, 2, 3, 2, 3.